Thank you for joining us for Metabolon's half-hour webinar series, where our topic today is Metabolomics, a powerful tool for basic plant science and agricultural biotechnology. We will have a short period of time for Q&A at the end of this webinar. Please feel free to submit your question via the Q&A section at the upper right corner of the screen. And just a quick reminder, this webinar is being recorded. Thank you. Your presenter for today is Dr. Danny Alexander. Dr. Alexander has over 30 years of experience in plant molecular biology and biochemistry. Before joining Metabolon, Dr. Alexander held positions at ARCO Plant Cell Research Institute and Calgene, working on cloning of agriculturally important genes, and later worked at Ciba Geigy, Novartis Syngenta, and BASF, working on plant disease resistance, maize molecular genetics, and genomics. Dr. Alexander has been at Metabolon since inception in 2003, initially working on development of the Metabolomics platform and software, and more recently, providing analysis and biological interpretation of study results. His scientific papers related to work at Metabolon have been published in leading journals such as Nature, Nature Medicine, Cancer Research, Plant Cell, and Plant Physiology, among others. Dr. Alexander currently serves as Senior Program Manager for Plant Science. I'll be turning the presentation over to Dr. Alexander now.
Thank you, Brandon. And thank you for joining the webinar this morning. I wanted to just say some words about how we use metabolomics and how it can be applied to plant science. Here at Metabolon, we've done approximately 450 projects related to plant science, which is a minority of the over 5,000 that we've done, including all the medical sciences. While it's still a lot of projects that we've done, about half in the academic sphere and about half in the commercial sphere with the agricultural biotechnology companies. So I wanted to address today, just start out with the kinds of studies and the kinds of targets that we can apply metabolomics to, and these are basically a list of things that we have applied it to in projects that we've run in both academic and the commercial spheres. Among these are many biomarkers for various traits in the ag biotech world, or yield and heterosis, and then the quality of either fruit or the crop quality, plant health, that sort of thing. We've done several in various stresses, abiotic and biotic stresses, and metabolomics is really good for finding the effects of mutants and transgenes, and you can use it for transgene analysis, whether doing equivalent sort of tests to compare to a wild-type plant, or if you've engineered a pathway for altering performance, then there's a lot of uses there to see for either event selection or the quality of the performance of a particular trait. We also applied it to microbiome type projects for using strain evaluations for microbiome and how those microbes interact with plants. We've done germplasm diversity studies and a fairly large number of developmental things, especially development of seeds, plant development, early growth, that sort of thing. And we've worked on chemical toxicity studies, but also mode of action for agricultural biochemicals. And there's others that aren't covered on this list, and maybe others that we haven't done yet, but we could certainly approach. But the core of it is to have a biological question that everything is framed around. So let me just tell you first the technology. I'll just briefly cover the technology for the Metabolomics platform. We call this the HD4 platform. It's the fourth generation, and it also uses 4 UHPLC platforms or data streams with different conditions, which cover a range of polarities, different ionization properties, and these 4 streams are really designed to maximize the number of compounds that we can detect and put in a data set. So basically we take one sample, do an extraction with methanol water, and we split that into 4 parts and then run it on these 4 different platforms, and each one generates a very large number of ion features. And the real, I think the thing that has been a key to our success really is the software. We have been, a very large amount of our effort goes into software development. The mass specs and the chromatography and all that is really off-the-shelf stuff, and it's, a lot of those methods have been published. The real proprietary and important part that we have at Metabolon that makes it work is the software that will eliminate that noise, reduce that data down to just signals for known compounds that we are sure that we can call. We do this by what we call a library search method. We establish library entries for over 4,500 purified standards, and we know how those chemicals perform on all 4 of these platforms. We know all the associated ions, the MS fragmentation patterns, and so on. And so all that information constitutes a library entry, and we run a sample. We scan that data for all those library entries and find the matches in that case. And we don't put a compound in a data set unless we're sure of the identity of that compound. And in plants, we typically lately have been reporting about 500 to 600 compounds in leaves, roots, and things like that, and depending on the matrix somewhat. Seeds may have a bit fewer, but let's say 500 to 600 compounds roughly is what we've been finding. So we take that data, that raw data now, and reduce it down to a data set. Is essentially 1 compound per sample is reported in a semi-quantitative fashion. We do statistical analysis to understand the changes in that data in relation to your study design, which is related to whatever the question is that you're asking, whether it's the effect of a mutation, the effect of a stress, so on and so forth. We do that statistical analysis and display it in various ways, various tools that we have to display the statistical results in table form, in sort of a classic, fold increase, fold decrease sort of thing. We can project those onto maps, which I'll show you an example of later, how those results are displayed. The compounds are classified by the general biochemical pathways and association types. And then, more importantly, sort of the main product that we offer that I think that other people don't is that we take those statistical results and we provide an interpretation based on what we can learn from the literature or what we already know, or from our institutional knowledge of running thousands of studies. We can make an interpretation, at least a high-level interpretation of that, the biological importance, the metabolic ramifications of the study design that you're talking about, and provide then the biomarkers and mechanistic understanding and so on of your data set. This is a representation of, it's a posterized version of the plant pathways that we can display our data into. In this particular layout, the blue dots represent compounds for which we have library standards and for which we can search. If that compound is in a high enough concentration to be detected, then we can report those compounds in your data set, and then those results, statistical results, could be displayed in this environment. This is a version with the poster stuff stripped away to show that the various areas of metabolism that we cover. Plants are more anabolic in nature. The animal version we have of this is primarily catabolic. So we have two general environments. This one is for the plants, and so it's go all the way from the carbon assimilation area, the energy pathways, biosynthesis of all the amino acids, and several secondary metabolites. So I'll show you an example or two of that later, some data in it. So at this point, I'd like to just share with you, I think, 3 studies, just a small amount of information from 3 different studies that we've had. These are published examples, or at least the data is publicly known for some of these, and I'll just sort of briefly go through those, and then we'll let you submit questions if you have questions for that. So the first example I wanted to go through is a study we did with Dr. Raju Datla up in Saskatoon. He wanted to study the TOR pathway in plants. So TOR stands for target of rapamycin, and rapamycin is the reagent that's been, very useful for elucidating the properties of all the different regulatory proteins and pathways involved or controlled by the TOR complex. TOR complex is really a central integrator of growth processes, energy flow, and growth in all eukaryotes. But the problem was in plants that rapamycin, it was not a target of rapamycin per se, because one of the subunits did not make. I'll show you a picture of that. So the rapamycin subunits, the pocket is formed by the mTOR itself and the FKBP12 protein. Well, the FKBP12 in plants has a different conformation, and so the pocket does not form, so rapamycin doesn't inhibit. So they engineered Arabidopsis to express a yeast version of this protein, and then it conferred on that Arabidopsis the ability to be inhibited by TOR. So then they did these studies of the effects of rapamycin by inhibition of the TOR pathway. So when they inhibit TOR in Arabidopsis, you get many effects that you might expect, and a sort of a starvation response in the plants. The roots are shorter, root hair elongation is smaller, the leaves and cells are smaller, and it delays development in general. The flowering is later and senescence is later. It's equivalent to a caloric restriction in animals, and in fact, if you give rapamycin to mice, it extends their lifespan somewhat, and it's sort of a caloric restriction, sort of right on the verge of starvation. They live longer. It's not pleasant, but it's one way to extend your life, is to have it very restricted. And so this is a similar response in plants. You get delayed senescence in plants also. Now the metabolic effects of TOR, this is just a very high overview of the data that we saw, the effects that we saw. On the left are the wild type and the 2 transgenes. These are 2 events of the same type of the transgenic plants, and so you see on the left where they're not treated, there's not much difference. You can't really see any differences between those. On the right, when you treat with rapamycin, you see that the wild type is the same as the untreated, and then the 2 events that are susceptible to rapamycin inhibition have very extensive metabolic perturbations. And so rapamycin, first thing it does is inhibit protein translation. So you see a higher levels of free amino acids and other metabolites are involved in a lot of anabolic processes, such as polyamines. Dipeptides are lower because you're not getting the protein turnover. There's increase of activity in the glycolysis and the TCA cycle, because this signal is essentially a starvation response signal. And so those pathways are demanding more energy, and they do that at the expense of the storage oligosaccharide pathways, among others. We also see increase in synthesis in the glutathione pathway, which provides the antioxidant capacity to fight oxidative stress, which is brought about by this inhibition. We also see accumulation of cell wall precursors because the cells are not dividing as rapidly as they were. So those are just the general overviews. One pathway, if we look just at the energy pathways here, so it remodels the carbon distribution to feed more photosynthate and to draw on carbon stores, in this case raffinose we show, to feed into the glycolysis and the TCA cycle. So you see dramatic increases in a lot of compounds in those pathways. And also, as a sort of a side note, Dr. Datla did RNA transcriptomic analysis of these same samples, and one of the largest a very large inhibition of the galactanol synthase gene, which is the regulatory gene that controls flow of carbon into the raffinose oligo pathways to be used as storage. And so it's diverting photosynthase directly, and it's drawing down on the stores of raffinose in this case. This is that same data essentially displayed in the cytostate pathway statistical display environment that you saw on the large map. The large map had all the pathways we have, and then we have a series of smaller maps, individual pathway maps that are linked by these additional links. So you can go from subpathway to subpathway by clicking on links like this, or you can move around inside the big map itself. But the results are displayed on the compound nodes. If the red node indicates increase in the compound, and the blue indicates decrease, and the magnitude of the change is reflected in the size of the dot and so forth. This is a very customizable kind of display. You can add on back in the all the minor metabolites, the cofactors, and all that sort of stuff. So it's a very useful environment in which to explore the results, and that's available on our portal, which when the project is delivered, you get a link to that portal and an account to display your data and to dig through your data in that environment. So the next example I'd like to cover is a study we did with Dr. John Cushman, who's at the University of Nevada in Reno. John's an expert in abiotic stress and drought and that sort of thing. He'd been studying ice plant for several years as a model for sort of diurnal carbon management. So it turns out ice plant is a facultative CAM plant. When it's under drought stress, it uses CAM metabolism. But he had a starch mutant, a full mutant that could not make any starch, yet the plant grew quite well. So the question was, this phosphoglucose mutase mutant grew quite well, but it made zero starch. So the question was, how did the plant manage its daily carbon input and output, because it was only taking in CO2 at night. So the study is a bit complicated. He has the wild type and the CAM deficient mutant together. They did both water stress and unstressed. So there were the well-watered plants, and then there was a drought period to induce the CAM metabolism. And he took, over a period of 3 days, he took samples every 8 hours. The first day he had a full day-night cycle, and then he went straight for 2 days of constant light. So he was really looking, also asking the question, with the compounds that were affected, how many were working in sort of a circadian rhythm. So getting straight to the sort of the main, well, I take it back. Here's an overview, basically, of the way the data look in those two from a high altitude. You'll notice that the wild type and the drought stress of the wild type do show a lot of metabolomic changes, a lot of effects there, many of which appear to be diurnal or circadian in nature. The mutant itself is in the well-watered plants. It's quite different from the wild type, and the drought-stressed mutant is obviously much different from the unstressed mutant and from the stressed wild type plant. So there's a lot of things going on there in terms of carbohydrates and lipids especially. And so the key question, I guess, was how does this starch mutant manage its carbon in a diurnal way if it can't make starch? And so the bottom line is that it uses lipids and also sugar alcohols to do a lot of this. So up on the left, you see that the red lines are the mutant itself and its essentially flatlined for, in this case, maltotriose, but all the maltodextrins, and for starch. There were no measurable starch in these plants, and in the wild type plant would make starch, as you might expect, sort of diurnally, and then in the constant light it accumulates more starch. But in the mutant, which are the red and the pink lines, and the pink line is the mutant under stress, and so what you see is that it accumulates some sugar alcohols, some inositol metabolites, as well as several lipids, fatty acids, and these monoacyl glycerols. In this case, they pretty much follow the pattern you'd expect for starch, except they appear to be more circadian in their responses in this. But this is being looked at as sort of the way that the plant manages its daily carbon budget, and remains able to grow even without being able to make starch. Okay, so quickly, the last example is a fascinating study done by Dr. Mel Oliver at the University of Missouri. Mel studies resurrection plants. In this case, he has two sister species of Spirobolus, which is a grass taken from Africa. One is desiccation sensitive, and the other is desiccation tolerant, which is to say it's a resurrection plant. And so we wanted to compare those, sort of ask three questions. How are they different at the start? How do they respond once you start taking water away? And then what are the late stage effects that you see in the desiccation tolerant plant as it dries down all the way to dryness and maintains its viability? In the next slide, I just will remind you that drought and desiccation are much different things. Drought tolerance is some plants are drought tolerant, but they can only do a temporary management of suboptimal water conditions. They accumulate osmolytes and adjust their carbon-nitrogen balance and all. But once you lose too much water, they inevitably will die. Desiccation-tolerant plants is the ability for the vegetative tissue to go to complete protoplasmic dehydration and then be able to survive just by the readdition of water. And to remind you that is a pathway and it's a process that a lot of plants do, but only in seeds. So normal seed desiccation in orthodox seeds is actually the same program. If you look at the accumulation of the osmolytes, the accumulation of antioxidants, and various other processes in a very controlled, very regulated manner. The trick is that the resurrection plants can do this in vegetative tissues. So they've taken that pathway now and reactivated in vegetative tissues. That's the definition of a resurrection plant. Very briefly, at the initial state, the resurrection plant, Staphyonus, is already essentially, in quotes, pre-induced for things that are rich in nitrogen and in osmolytes such as sugar alcohols, and especially glycerophosphocholene, which is a very powerful osmolyte actually that the kidney uses to protect cells from very strong salt and urea concentration. So it's one of the most powerful osmolytes known. In the initial dehydration response, just going from 100% to 60% hydration, you see that the Staphyonus mounts a very strong response to produce osmolytes, antioxidants, oligosaccharides, and such in order to further prepare it for protecting the cells during dehydration, whereas the remodels, the susceptible line, essentially does nothing. It just does not have that kind of response. That pathway is totally absent, essentially. And in the late stage desiccation is dedicated to accumulating a lot of antioxidant compounds. And the glutathione pathway that synthesizes glutathione accumulates a lot of gamma-glutamyl amino acids, which are just intermediates in the pathway, but it turns out probably to be very powerful osmolytes themselves. You accumulate glutathione. You also accumulate the tocopherols for antioxidant purposes. So all this It tends to make sense for ways that the protoplasmic dehydration can be tolerated in these plants. So that's the three sort of examples I wanted to show you. And if you're considering a metabolomic study in plants, I just wanted to go over a couple of things to remember and things to consider. And the main thing to ask is, what's the question? What's the problem you want to solve? It needs to be framed in terms of the biology of your system. And if it's not really a biological question, if it's like if you're looking for a new chemical or something like that, maybe metabolomics is not the right approach. So if you want to approach the questions of in a biological manner, and so then we need to consider statistical power. How reproducible is the phenotype? You need the shaky or the sort of the wobbly phenotypes require more biological replicas in order to have good statistical power. Things that have a very tight phenotype you can get by with very few reps. And then that raises the question, what is a biological replicate? And it's not, two different leaves from the same plant. It has to be, to make the good study design, you want to, in the case of soybeans over here, you wouldn't use different leaves from the same plants and call those replicates. You'd have to have the same leaf stage on independent plants treated the same way to make a group. And also to remember that everything's a gradient and a cycle. So a leaf from base to tip, a leaf is very different metabolically. It's a big gradient for a lot of different compounds, and the time of sampling is very important because everything operates in the circadian manner and diurnal manner because of the light-dark cycles. And so you've got to keep those kind of things in mind when you design a study design, how it's executed, when and how the samples are handled and operated, and so on and so forth. So we could look at some questions that we have so far and approach some of those if you can. Okay, so one of the questions we're getting is says, can this be done non-destructively and using volatile organic compounds? Well, the answer for both of those is no. It is destructive. You do a methanol extraction of the sample to crash out proteins and collect the methanol soluble fraction, and it's all done in ambient, so there's no volatiles. Our system essentially dries down the sample and so on, and so the volatiles will all come off, so that would not really apply to that. Another question we've got here is how much sample is needed and how is it prepared? So the amount of what I really recommend for plant tissues is that they be harvested into liquid nitrogen and ground up into a powder and then lyophilized. That gives, so not only does it make it easier to handle sample, but let's say if you harvest a whole leaf, if you take the tip of that leaf versus the base of that leaf, you get a different result. So either you've got to grind up the whole leaf and average all that out, or take a particular piece of the leaf, and it's better to grind that up to make a uniform sample. Another part of that question was about the photosanitary requirements. If samples are from the U.S. and being analyzed here in the U.S., there are no sort of requirements or permits or anything. However, if samples come from, say, Canada or from Europe into our country, and if there are, if it's, say, a plant disease study and there are residual pathogens on that plant, then a USDA permit is required for that, and it takes a few weeks to get that. Let's see. We have another question. Okay. Another question that came up It's about identifying the compounds. I think I addressed this earlier. The way we do metabolomics is called chemicentric. That is, we look for chemicals. We don't analyze the ion peaks per se. Our library entries sort of accumulate all the ions, the ratios of those ions, the MS-MS fragmentation patterns in order to identify a compound, and then one peak, the integrated area under one peak, usually the largest peak, or at least the largest unique and descriptive peak for that, is integrated to provide a relative quantitation for that compound. And so the identity is known at that point. Almost all our calls are what are called Tier 1 calls. The chromatography, the accurate mass, and the MS-MS fragmentation pattern have to all be accurate or have to all agree with our library entry in order to make that call. If we don't get all three of those parameters met, we typically just leave that compound out of the analysis. So it's not, it doesn't pass our QC. So if there are any other questions, we need to wrap up, I guess, based on time. But if there are any other questions, feel free to email us or me directly, if you like, with any questions you might have. And if you'd like to contact us more about conducting a study or what a study might look like or costs and things like that, and we'd be glad to address that. So thank you again for your time. Glad you joined us. I hope you found it useful and interesting.
Thank you for attending our webinar. If you have an additional question, please email us at badams, B-A-D-A-M-S, at metabolon.com, and we'll get back to you shortly with an answer. If you'd like to share the replay or listen again, the recording will be available soon on our website at www.metabolon.com.